Hello everyone, we are Group 6, and today we are presenting about gold nanowires and nanoparticles. The people that are in Group 6 are called Matthew Jimenez, Thomas Shoff, and Naomi Pineda. The role of gold in society has largely been for jewelry and other applications. Humans' fascination with gold in medicine has been going on since the Middle Ages. Colloidal gold was first studied in alchemist laboratories and was tested as a panacea with no real success. The first applications of gold colloids to biological studies occurred in 1971, and future studies have identified gold nanotechnology as an important area of research to target. Over the past 20 years, nanowires have become crucial within the medical and environmental industry. The reason for this being is because nanowires contain unique properties like atomical, electrical, and photothermal. These properties allow nanowires to have a variety of use within these fields. The optoelectronic effects cause nanowires to have catalytic reactivity. This makes nanowires crucial for purification processes within water. Nanowires are able to remove the contamination in water due to their physical absorption abilities like degrading contaminants through catalytic reactions and detecting the contaminants within the water sources. Some of the important contaminants that nanowires can detect is metal. This is significant because it could detect and remove any potentially harmful contaminants. Nanowires are able to remove these heavy metals through an ion exchange, adsorption, or amalgamation. Nanoparticles can also be useful within the biological field. Nanoparticles are utilized within the biological field to detect all types of metal ions, anions, small organic molecules, oligonucleotides, and proteins. Nanoparticles can help detect the pathogens and cancer. This is done through electrostatic interactions between the AUNP, or otherwise the nanowire and nanoparticle, and the targeted pathogen or cancer. AUNP are revolutionary because they could utilize microbalance-based sensing for detection of oligonucleotides and proteins. These different processes are revolutionary within the bioindustry because they can detect and remove unwanted pathogens or cancer. This is a huge step in today's society because AUNPs could potentially become one of the top potential cures for cancer. Cysteine-modified AUNPs could also detect small organic molecules through aggregation. If a reaction occurs with the presence of a cysteine-modified AUNP, then this confirms the presence of a small organic molecule, and this could also determine the presence of an explosive molecule. Okay, so now moving on to a more depth analysis of the production of gold nanowires, here's Naomi. Take it away, Naomi.
One of the methods used to create gold nanowires is by using gold 3-allylamine planar complexes and triisopropylsilane. At first, gold 3 ions form complexes with a planar square geometry and are then gradually reduced to gold 1, forming disc-like structures by the aggregation of gold 1 and/or gold 3 complexes. Continuous stacking of disc-like objects leads to the formation of a cylinder. After most of the gold 3 ions are reduced to gold 1, the reduction of gold 0 starts. The average diameter of the object is reduced and the length is increased while forming a hexagonal arrangement separated by a bilayer of gold 3-allylamine planar complexes molecules. The length of gold nanowires continues to grow during this slow reduction of gold 1 to gold 0. Finally, bundles of gold nanowires with the lengths of several microns are obtained as a final product. Due to the optical properties of gold nanoparticles, the UV spectra is both size and shape dependent, meaning that depending on the size and the shape of the nanoparticle, we would see different color solutions at different wavelengths. Now, why are gold nanowires important?
Now that we've learned the basics of nanoparticles and the process by which gold nanoparticles and nanowires are made, let's take a look at some of the uses for these amazing particles. Due to their unique physical and chemical properties, gold nanoparticles have been targeted by researchers for a variety of uses in medical technology, chemical testing, and a variety of other fields. One of the more exciting areas of research here is in connection to gene therapy. In a 2013 study, researchers used gold nanowires as the basis of a nanoinjector that allowed them to deliver both linear and plasmid DNA directly to cells. The advantage of using gold nanowires for this technology is their electrical properties and their high level of spatial selectivity. First, a DNA molecule is attached to the gold nanowire injector, and the injector is inserted into the cell by piercing the cell membrane. When an electrical impulse is sent through the gold nanowire injector, the DNA molecule releases, allowing for specific timing of release of the DNA. Due to the small size of the gold nanowires, they are able to pierce the cell wall without doing significant damage to the cell structure. This is a key advantage that prevents cells receiving gene therapy from undergoing apoptosis or dying due to the injector piercing their cell membrane. These properties together show the incredible value of gold nanowires as gene delivery devices, but this technology can also be applied to vaccination, targeted drug delivery, and understanding of cellular events. Gold nanoparticles and nanowires have also been applied to a key environmental concern, contaminated water. One use of these gold nanoparticles is to act as a chemical sensor and detect different kinds of toxins in the water. Sensors like this have been developed for heavy metals, pesticides, and bacterial toxins. For heavy metal sensors that detect things like mercury, chromium, and lead, gold nanoparticles are generated that are capped with quaternary ammonium group terminated thiols. These thiols react with heavy metal atoms present in solution, particularly with mercury, causing the freed gold nanoparticles to aggregate in clumps. This grouping causes the suspension color to change from red to blue, showing the presence of mercury atoms in solution. This same concept, known as a colorimetric sensor, has been applied to lead ions and chromium ions as well. Gold nanoparticles have also been used to purify water by removal of dissolved organic compounds. To facilitate this, gold nanoparticles are composited with polydimethylsiloxane, a silicon-based polymer, to create a porous foam containing the gold nanoparticles. This composite has the ability to absorb a variety of aromatic compounds and hydrocarbon oils. One such application of this composite is the removal of diesel from water, which is quite difficult to do by most other methods since water and diesel form a stable emulsion. To observe this ability in action, researchers first filled a petri dish with water. Then, some diesel is added to the water and the gold nanoparticle foam is prepared. When the foam is placed into the dish, gold nanoparticles in the foam coordinate around the diesel, allowing the foam to absorb the diesel. Once it has fully absorbed the diesel, the nanoparticle foam is then able to be filtered out of the water, completing the purification process. With their incredible potential to make change across a variety of fields, gold nanoparticles and gold nanowires are primed to become one of the compounds that can change the world. Thank you so much for listening to our presentation.